Look at the footprint of a standard psychiatric biomarker laboratory. It takes entire rooms of equipment and specialized technicians to process a single sample. Now imagine compressing all of that massive infrastructure into a sleek box that sits comfortably on a doctor's desk. This is the exact problem the SC meter is designed to solve. It is a prototype instrument engineered to run a very specific 18-gene mRNA biomarker panel used for psychiatric diagnostics. To make this work, the hardware has to execute a complex sequence of chemical extractions and rapid thermal amplifications, entirely automated within a single, highly constrained physical footprint. Engineering the thermodynamics and fluid routing is the first requirement. The second is aligning that physical work with a specific legal architecture. The device's physical components must be precisely mapped to intellectual property boundaries to ensure the inventor has the freedom to operate in the commercial market. This mapping relies on a strategy known as stacked IP. It involves dividing the product's innovations into three distinct separate layers. The foundational layer is the underlying biological science. In modern diagnostics, the rights to the specific genes being tested are often owned by a third-party research institution. The middle layer is the utility patent. This protects the novel physical architecture, the valves, pumps, and thermal controls that the engineer actually developed. The top layer is the design patent. This covers the non-functional visual appearance of the graphical user interface, how the final data is presented to the physician. Clearly delineating these three layers, identifying what is licensed, what is functional, and what is aesthetic, is what allows the instrument to exist as a viable product. When designing the SC meter, the engineering team had to build around a locked operating environment. They had to create a machine that executes a specific biological test without altering any of the core science. Inside the SC meter, that core science is an 18-gene biomarker panel developed by researchers at UCI and Laguna. The identity of those 18 specific genes, along with the mathematical classifier coefficients required to interpret the results, are completely fixed. That algorithmic framework is already fully protected under pre-existing patents, specifically US 11713-486b2 and US 12404-553b2. Because this diagnostic core is locked third-party intellectual property, the inventor of the physical machine claims absolutely zero ownership over the medical conclusions it generates. Their territory begins exactly where the biology ends. That territory starts at the front end, where the hardware must take a raw biological sample and prepare it for analysis. The first step requires mixing the sample with a precise chemical buffer to strip the viral RNA away from the surrounding cellular material. Once stripped, microscopic magnetic beads are introduced into the fluid flow to physically capture and isolate that specific RNA payload. The mechanical challenge here is incredibly severe. The device has to route exact microvolumes of these reagents through a closed circuit without any risk of cross-contamination. To handle this, the system relies on specialized pressure and flow controllers to push the fluids with extreme physical precision. Mux valve controllers and droplet generator chips then act as the physical traffic directors, ensuring the chemical components arrive at the correct location at the exact right millisecond. Integrating these specific off-the-shelf fluidic and chemical components into a seamless automated loop forms the first pillar of the inventor's novel utility claim. After the mRNA is isolated, the instrument must immediately process it through rapid thermal cycling. This requires high-performance thermoelectric hardware, like a specialized Peltier plate, capable of aggressively heating and cooling the sample block. A dedicated PID controller manages these thermal shifts, maintaining the extreme temperature tolerances the RNA needs to successfully amplify. With the sample amplified, the system moves to the optical detection phase. The machine now has to physically read the results of the biological reaction. It achieves this by firing targeted 590 nanometer LEDs at the sample, exciting the molecules and triggering a fluorescent response. Highly sensitive photomultiplier tubes, or PMTs, are positioned to capture this microscopic fluorescent light. These tubes serve as the crucial bridge, translating physical photons into the structured digital data that the third-party biomarker algorithm needs to run its calculations. Architecting this continuous sequence, from fluid extraction through precise thermal manipulation to final optical detection, completes the engineering requirements necessary to file the hardware utility patent. Now we step out of the internal mechanics and look at the external user interface. This is where the physician actually interacts with the finalized data. The device utilizes a commercial 7-inch touchscreen to display its graphical user interface. However, the layout, typography, and visual flow of this interface do not perform a mechanical function. Therefore, they cannot be covered by the utility patent we just outlined. Instead, the visual presentation is protected by a design patent application. This specific legal tool secures the ornament and aesthetic appearance of the software, isolated from how the underlying code actually works. The design patent acts as the final protective shell. It locks down the specific visual identity of the SC meter, completely independent of its internal hardware operations. We can now view the entire machine as a completed intellectual property map. The biological algorithm evaluating the sample is tagged as a third-party asset. It is strictly licensed IP. The fluidic, thermal, and optical hardware orchestrating the test is the inventor's proprietary domain. This is protected by the novel utility patent. And the final graphical interface displayed to the user is secured under a dedicated design patent. The resulting architecture demonstrates a strict separation of domains, where the proprietary hardware functions independently of the licensed biological logic. This highlights the ultimate reality of point-of-care medical device development. Inventing the box does not mean you own what happens inside it. No matter how sophisticated the internal fluidics or thermal controllers are, commercializing the SC meter strictly requires an active license for that 18-gene panel. But this modular legal strategy protects the hardware engineers. By keeping the boundaries clear, they retain the option to license their utility architecture to run entirely different biological tests in the future. Bringing a complex diagnostic tool to market demands rigorous physical engineering. But without an equally rigorous strategy for mapping out intellectual property, even the most advanced machine will never leave the laboratory.